Hello everyone, my name is Nur Akila binti Abdul Paro and this is my final year project presentation. The title of my research is Molecular Identification of Bacillus cereus isolated from agricultural soil sample. In this presentation, I will walk you through the background of the study, objective, methodology, results, and my final conclusion and recommendation. This is the overview of my presentation. It will begin with the background of the study, followed by the problem statement and research objective. Then, I will explain the methodology used to conduct this research, present the results and discussion, and end with a conclusion and recommendation based on my findings. Bacillus cereus is a type of gram-positive, root-shaped, spore-forming bacterium that is commonly found in the soil environment. Due to its ability to form endospores, it can survive in harsh conditions. Some strains of this bacterium are beneficial to agriculture as they help promote plant growth. However, other strains produce harmful toxins that can cause foodborne illness when they contaminate crops or food products. For this reason, identifying and monitoring Bacillus cereus in agricultural soil is very important, especially for ensuring food safety and public health. Although Bacillus cereus can play a positive role in agriculture, the presence of toxic strains poses a serious risk. Soil is a complex environment filled with diverse microorganisms, and Bacillus cereus can be one of the dominant species, especially in agricultural settings. If the soil is contaminated with harmful strains of Bacillus cereus, it can lead to crop contamination, which may eventually enter the food chain. Therefore, it is important to detect and identify this bacterium to assess any potential risk to human health and food safety. This research aims to achieve two main objectives. First, to identify the presence of Bacillus cereus from soil samples using biochemical tests and morphological observations. And second, to confirm the presence of Bacillus cereus isolates through molecular identification using the polymerase chain reaction or PCR technique. The methodology involves several stages. First, soil samples were collected from agricultural land at a depth of about 5 to 10 cm below the surface. These samples were diluted in sterile saline and cultured on strip-tip soil agar to promote bacterial growth. Colonists were then observed for their morphology and gram staining was performed to examine their cell wall characteristics. Biochemical tests such as catalase test, starch hydrolysis test, and casein hydrolysis test were conducted to assess enzyme production. DNA was then extracted from selected colonies. PCR was performed using universal 16S rRNA primers, 27F and 1343R to amplify the gene segment for further analysis. In this step, bacterial colonies were isolated from the soil dilution series. A positive control, which is a non-Bacillus cereus strain, was used for comparison. The morphology of the soil isolates was compared to the control. Colonies labeled S21 to S25 were selected for further testing because they show growth patterns similar to Bacillus cereus, including clear, spreading edges which are typical for this species. These colonies were then subjected to gram staining and biochemical tests. The gram staining results for the soil isolates show purple, root-shaped bacteria under 1000 times magnification, which is consistent with gram-positive bacteria. This matched the morphology of the positive control. The observation of root-shaped cells confirmed that the isolates had structural similarities with Bacillus cereus, which is a critical first step before confirming the identity through biochemical and molecular testing. In the catalase test, both the positive control and soil isolates produced visible bubbles when hydrogen peroxide was applied. This indicates the presence of catalase enzyme, which breaks down hydrogen peroxide into water and oxygen. The production of catalase is a trait associated with aerobic bacteria like Bacillus cereus. The presence of bubbling in the soil isolates supports the hypothesis that they belong to the same genus. Next is the casein hydrolysis test. In this test, clear zones were observed around the bacterial colonies on skim milk agar, indicating the breakdown of casein protein. Both the soil isolates and positive control produce these clear halos, showing proteolytic activity. This means that the isolates can produce caseinase, which is a protein-degrading enzyme, and this further supports their identification as Bacillus cereus. In the starch hydrolysis test, iodine was added to the ECA after incubation. The salt isolates, like the positive control, show clear halos around the bacterial growth, indicating that starch was broken down by an amylase enzyme. The ECA failed to turn blue in the areas surrounding the colonies, showing a positive reaction. This again confirms the enzymatic activity consistent with Bacillus cereus. Polymerase chain reaction, or PCR, was carried out to amplify the 16S rRNA gene using the universal bacterial primers 27 forward and 1343 reverse. As shown in the gel electrophoresis image, clear DNA bands were observed for the positive control as well as for soil isolate samples labeled lane 1 and lane 2. The bands appear at approximately 1,300 base pairs, which is consistent with the expected product size. The negative control lane showed no visible band, which confirms that the PCR reaction was free from contamination. The presence of strong, clear bands in the sample lanes indicates successful amplification of the 16S rRNA gene, which is essential for further identification by sequencing. After PCR amplification, the products were sent for sequencing. The resulting DNA sequence was then analyzed using the NCBI BLAST tool to compare with known sequences in the GenBank database. The BLAST result showed that the sequence obtained from the soil isolate had 97.82% identity with Bacillus cereus sequences in the database. The high percentage match between the query and the subject confirms that the isolate bacterium is Bacillus cereus. This molecular identification step is crucial because it validates all previous biochemical and morphological tests, giving us confidence in the accuracy of our result. In conclusion, Bacillus cereus was successfully isolated from agricultural soil using morphological, biochemical, and molecular techniques. Gram staining and biochemical tests confirm it as a gram-positive root-shaped bacterium. PCR and sequencing further validate its identity. This study highlights the presence of potentially pathogenic bacteria in soil, which is relevant for both agriculture and public health. I recommend that future studies expand the scope by identifying other soil microorganisms to better understand the microbiodiversity present in agriculture environments. This will help determine if there are other harmful or beneficial microbes that may impact food safety or crop production. Additionally, regular monitoring of agricultural soil is crucial to reduce the risk of crop contamination and ensure public health through safer food production systems. I hope this presentation has provided clear insights into the methodology and importance of identifying soil microbes that could impact food safety. Thank you so much for watching.